Hello teacher, good day. In this presentation, I will explain generation of antibodies and some of their characteristics. Humoral immunity. What are antibodies or immunoglobulins? They are a family of glycoproteins produced by B lymphocytes in two forms, membrane and secretory. They are composed of two heavy chains identical to each other by covalent bonds and two like columns. They are composed of N-terminal variable regions and carboxy-terminal constant regions. The 5 regions contain 3 independent hypervariability regions that are especially assembled to form the antigen binding site. Response of B lymphocytes to antigens. Primary immune response. The antigen adheres to the receptor of the B lymphocyte. The B lymphocytes transform into memory cells or plasma cells with the help of T lymphocytes. The plasma cells produce antibodies specific for the antigen. The primary immune response is slow. The secondary immune response is memory B lymphocytes immediately recognize the same antigen, multiply, transform into plasma cells, and produce antibodies. The secondary response is fast and effective. Activation of B lymphocytes. Mechanisms of activation by independent antigens. Protein antigens cannot by themselves induce the activation of B lymphocytes, but require the simulation of CD4 plus lymphocytes. In this picture, the antigen receptor binding. The antigen is internalized and processed in endosomal vesicles. It is derived by enzymes. Peptides plus MHC2 molecules. The complex costimulatory proteins are expressed. They are found in the B lymphocyte membrane presenting the antigen to the LTH. Activation mechanism by independent antigens. They are polysaccharides, glycoproteins, and nucleic acids. These antigens are not internalized, but exert their activation by intracellular signaling produced by the B lymphocyte receptor. Generally, the response produced by this step antigen is made up of antibody with low affinity and a low repertoire of memory cells. The practical importance of this step or rectation is that many antigen in the bacterial wall are polysaccharides, and in this main mechanism for activation of B lymphocytes in immunity against bacterial infections. Transformation to plasma and memory cells. When a B cell encounters an antigen, it begins to mature into a plasma cell or memory B cell. How is the maturation process of B lymphocytes carried out? B lymphocytes begin their maturation in the bone marrow and complete in the peripheral lymphoid tissue. During this process, the lymphocytes acquire antigen receptors that are immunoglobulins made up of heavy unlinked chains. What role do plasma memory cells have? Plasma memory cells provide an effective defense to attack and destroy common pathogenic microorganisms in the environment with which they may repeatedly encounter. Production of antibodies by plasma cells. Every plasma cell is essentially a factory for making antibodies. As long as the antibody and the antigen are matched, the antibody marks the antigen for destruction. Tiny lymphocytes are cells programmed to respond to remember antigens. T lymphocytes learn to distinguish self from foreign antigens. Cells from a clone only produce a specific type of antibody. The body can produce a large number of different antibodies to fight against to antigens that the body is exposed to. Secondary immune response. If the antigen penetrates a second time, the faster, more intense, prolonged secondary immune response will be produced. There will be almost no latency period. More antibodies will be produced, and they will last much longer in the blood, even several years. This immunological memory is based on lymphocytes, which after the first contact with the antigen, some of them become memory cells, B or T lymphocytes.